Okay, let's unpack this. Imagine this. A guy in the U.S., Tim Fried,
Mm-hmm.
spent nearly 2 decades letting some of the world's deadliest snakes bite him.
Yeah, I read about this. Just incredible.
Over 200 mambas, cobras, taipans, the works.
Sounds absolutely crazy.
It does, right. But here's where it gets really interesting. Scientists now think his blood might hold the secret
The secret to what?
to a universal snake antivenom.
Wow. Universal.
Think about that one person's, incredibly bizarre choice, potentially saving countless lives.
It's fascinating, the sheer improbability of it all.
Oh,
And the scientific implications are huge.
Right.
Because right now, if someone gets bitten, the treatment often needs to be really specific.
For that exact snake.
Exactly. For that type of snake.
Yeah.
Fried's case, well, it suggests his immune system somehow figured out how to fight off a huge range of venoms.
It truly is, like the researcher said, unparalleled.
Unparalleled, yeah.
Exactly. So for this deep dive, we're going to explore the tricky world of snake antivenom. We'll try to uncover why creating effective treatments is such a monumental challenge, and see how Fried's seemingly wild experiment might just offer a ray of hope. Because it's easy to overlook this, but snake bites, they're a massive global health crisis.
Oh, absolutely. If you connect this to the bigger picture, it's quite shocking, really.
How big are we talking?
Snake bites kill hundreds of thousands of people every year. Hundreds of thousands.
That many. Wow.
And leave many more with permanent disabilities. The WHO, the World Health Organization, they even classify it as a neglected tropical disease.
Neglected, so it doesn't get the attention it deserves.
Pretty much. This isn't some rare thing. It's a major problem, especially in poorer countries.
Yeah.
And our current antivenoms, they have serious limitations.
Right. So our mission today is to understand why it's been so incredibly difficult for scientists to develop these, broadly effective snakebite antibodies through normal means.
The conventional ways, yeah.
And what makes Tim Fried's situation so unique and potentially, revolutionary? Let's start with the venom itself. It's not just one simple poison, is it?
Not at all. Not even close. Think of snake venom as this incredibly complex cocktail.
A cocktail?
Yeah, like a master mixologist decided to throw in dozens of different dangerous ingredients. Experts say it can contain anywhere from like 20 to 50 different types of proteins, peptides.
50.
50, yeah. And even other things that help keep the poison stable. And each of these toxins, they can target different parts of your body.
Causing different kinds of damage.
Exactly. Very different kinds of damage.
Like what? Can you give us an idea? Paint a picture.
Well, some toxins, they're called hemotoxic toxins. They attack your blood.
Your blood.
And make it unable to clot properly. So you get severe bleeding internally and externally.
Oh, bad.
Very bad. Others are neurotoxic. They mess with your nerves.
Nerves.
Yeah, including the crucial ones that control your breathing.
Yeah.
Which can be, fatal pretty quickly. And then you have cytotoxic toxins that essentially cause your cells. Well, they cause them to break apart. Tissue destruction.
So it's like a multi-pronged attack hitting you all at once.
Precisely. Blood, nerves, cells.
Yeah.
It's brutal.
That sounds absolutely terrifying.
Yeah.
And to make things even more complicated, I heard that even if you're bitten by 2 snakes of the same species, the venom might not be identical.
That's right. It's another layer of complexity.
How does that happen? Can it change based on, what the snake has been eating?
Precisely. A snake's diet, its location, maybe even the time of year, it can actually influence the specific toxins present in its venom.
Wow.
So this incredible variability, even within a single species, it's a huge obstacle when you're trying to create a single antivenom that will work everywhere.
It's like trying to build one key for a million different, constantly changing locks.
That's a great analogy. So the core challenge, really, in a nutshell, we're not fighting one poison. We're fighting a constantly shifting arsenal of dozens of different attacks happening all at once.
Okay, so the venom itself is a constantly moving target. That really highlights why creating a universal antivenom is such a holy grail in this field.
Absolutely.
But let's talk about how we currently make antivenom. It's not exactly a straightforward modern pharmaceutical process, is it? It sounds a bit old-fashioned.
You're right. It kind of is. For a long time, and still largely today, antivenoms are produced by injecting animals.
Animals, like lab rats.
Usually larger animals, like horses or sheep. They get injected with small, non-lethal doses of snake venom.
Okay.
The animal's immune system then kicks in, recognizes the venom as foreign and starts producing antibodies to fight off those toxins.
So we're basically tricking a larger animal's body into creating the medicine we need for humans.
That's a good way to put it, yeah. We harvest those antibodies from the animal's blood serum.
Huh.
But the issue is, the resulting antivenom serum contains a whole mixture of antibodies, not just the specific ones that are effective against the snake venom.
Ah, okay. So lots of other stuff in there too.
Exactly. And this is where a lot of the limitations come from. Ultimately, the traditional method is a bit like using a blunt instrument.
Against a really sophisticated threat.
Yeah, against a highly sophisticated and variable threat like venom.
So what kind of limitations are we talking about? I mean, it's what we have, so it must work to some extent, right?
It does work, absolutely.
Yeah.
It saves lives. But it's far from ideal.
How so?
For one thing, current treatments often need to match the specific snake species that bit the person.
Right, you mentioned that.
Which means you have to know exactly what kind of snake it was. But you can imagine in many real-world situations.
Yeah, if it's dark, or you're panicked.
Or you just didn't get a clear look. Identifying the snake can be nearly impossible sometimes.
And what happens if you guess wrong? Or use the wrong antivenom?
Well, that can be seriously problematic.
Yeah.
Even life-threatening.
Ow.
It might not effectively neutralize the venom at all. So the person continues to, suffer the effects of the bite. Wasted time, wasted treatment.
And you're still injecting them with stuff.
Exactly. You're still injecting foreign animal proteins into their body, which can trigger severe adverse reactions in some people. Allergic reactions, serum sickness.
Okay, so identifying the snake is key, but often really hard. I vaguely remember reading about a simple blood test used somewhere. Southeast Asia.
Yeah. You're probably thinking of the 20-minute whole blood clotting test.
That's it. What does that do?
It's a valuable tool in that specific region, yeah. It helps doctors differentiate between certain viper bites, which tend to mess with blood clotting.
The hemotoxins?
Right. And bites from elapids, like cobras and kraits, which are often more neurotoxic. It guides the choice of antivenom there.
But that really emphasizes how localized these approaches are, doesn't it? It's not a global fix.
It absolutely does. It works for those specific snake types in that area. It's not a universal answer. And even when we have polyvalent antivenoms.
Polyvalent, meaning they work against multiple types of snakes.
Exactly. Designed to work against multiple species common in a region. But even they still might not cover all the dangerous snakes found there.
Serious?
Yeah, there have been cases where people were given a polyvalent antivenom, but it turned out not to be effective against the specific snake that bit them because that snake wasn't covered well enough by that particular mix, or the venom was just too different.
So we're relying on treatments derived from animals?
Mm-hmm.
That might not be a perfect match for the venom.
Right.
And can even cause harmful side effects themselves.
Correct.
That doesn't sound very efficient. I also understand that these traditional antivenoms don't have a very high concentration of the actual venom-fighting antibodies we need. Is that right?
That's absolutely correct. It's another major drawback. Traditional antivenoms might only contain somewhere between, say, 5% and maybe 36% of the specific active antibodies.
Only 5 to 36%. What's the rest?
The rest is mostly other animal serum proteins, stuff that doesn't really do anything therapeutically against the venom, but can contribute to those side effects we talked about.
Wow, so you need a lot of it.
Exactly. This low concentration means doctors often have to administer very large amounts of the antivenom, huge volumes sometimes.
Just to get enough of the good stuff, the active antibodies, into the patient's system to actually make a difference.
Precisely, to neutralize all the circulating venom.
Which I imagine leads straight into significant issues with cost and just how easy it is to get these treatments.
You got it.
If you need huge quantities of this stuff and the way we make it isn't super targeted or efficient, it's probably not going to be cheap.
No, it's not.
Especially for the people who need it most urgently.
Exactly. And that's the tragic reality. Snakebite is largely a disease that affects people living in poverty, often in remote, rural regions of the developing world.
The places where access to healthcare is already difficult.
Absolutely. So making sure that antivenoms are affordable and available in these areas is critical. But the manufacturing costs of traditional antivenoms can be high. And the supply, well, in some regions, particularly sub-Saharan Africa, it's dangerously low.
Scarce. It creates this terrible cycle, doesn't it? The people most at risk often have the least access to effective, affordable treatment.
It's a huge public health challenge. A vicious cycle.
It really paints a sobering picture of the challenges involved. So, against this backdrop, this difficult situation with complex venom and imperfect solutions, we have the extraordinary story of Tim Fried and his rather unique approach.
Unique is one word for it.
He definitely wasn't following any established medical guidelines, was he?
Absolutely not. Over 18 years, he essentially conducted his own extremely risky, unsanctioned human experiment.
Enduring over 200 documented snake bites.
Over 200 bites, yes. And over 700 injections of venom from a vast array of highly venomous snakes. We're talking some of the most potent venoms on the planet. Mambas, cobras, taipans, kraits.
And I really want to reiterate for everyone listening, this is incredibly dangerous. Do not try this. Absolutely not. The risks are immense and potentially fatal.
A crucial point. Cannot stress that enough. Fried put his life in grave danger repeatedly, period.
But from a scientific perspective, the outcome has been remarkable.
It really has. Somehow, his immune system adapted. It developed the ability to recognize and produce antibodies against an exceptionally broad range of snake venoms.
Which scientists are calling extraordinary.
Truly extraordinary, yeah. That's the word they use.
And the research on these extraordinary antibodies has shown some very promising results, right? I recall hearing they've protected animals.
Yes. In laboratory animal studies, antibodies extracted from Fried's blood have demonstrated the ability to protect mice, for example, against fatal doses of venom.
And not just from one type of snake.
No, from a diverse range of snake species, venoms that would normally be lethal.
That's a huge deal.
It's a significant step forward because it suggests the very real possibility of developing maybe not one universal antivenom,
Right.
but perhaps more broadly effective treatments.
Like maybe one that works against most of the elapids.
Like cobras and mambas.
And another one that targets most vipers.
That's the hope.
Yeah,
That
yeah.
kind of broad coverage would be a game changer.
And I also remember the term polyclonal immunity being used in relation to his blood. Can you break that down for us in simple terms? What does that mean?
Sure. So poly means many, right?
Right.
And clonal refers to clones or types of antibodies. So polyclonal immunity basically means his immune system has learned to produce a wide variety of different antibodies.
Okay.
And these different antibodies can attack many different types of toxins found across various snake venoms.
So it's like having an army trained to fight lots of different kinds of enemies.
That's a good analogy. Instead of a force specialized in only one kind of enemy, like many current antivenoms, which might target only a few specific toxins from specific snakes.
His body has developed a much more comprehensive defense strategy.
Exactly. It's something current antivenoms really struggle to replicate effectively.
So scientists are now intensely studying his blood, trying to figure out exactly how his immune system managed this super broad recognition.
That's the million-dollar question, yeah. Researchers are actively analyzing Fried's blood samples. They're trying to pinpoint the specific antibodies that are so effective.
And key players.
Exactly, and to understand the biological mechanisms, the processes that allowed his body to generate this kind of wide-ranging immunity over time.
And the ultimate goal would be?
Well, the ultimate goal is to identify these powerful antibodies and then potentially replicate them.
Make them in a lab. Safely.
Precisely. Replicate them safely in a controlled laboratory setting using biotechnology.
That sounds like a much more reliable and, frankly, ethical path forward than relying on the continued, very risky actions of one individual.
Absolutely. It's about taking the unique biological insight gained from his situation and translating it into a safe, scalable solution.
And I also saw some mention of combining his antibodies with other types of drugs. Is that being looked at?
Yes, that's another promising avenue. There's ongoing research exploring combination therapies. For example, scientists are looking at using antibodies inspired by Fried's, perhaps these lab-made ones eventually, in conjunction with other drugs. There's one called verisplatib, for instance.
Verisplatib.
Yeah, it's a drug that targets a specific enzyme found in many snake venoms called phospholipase A2. It's shown promise in protecting against a broader range of snake venoms, too.
So hitting the venom from multiple angles.
Exactly. This kind of multi-pronged approach, combining different types of neutralizing agents, could represent the future of snakebite treatment, making it much harder for the venom to win.
It's truly remarkable how such an extreme personal undertaking, however dangerous, could potentially unlock such significant benefits for global health.
It really is.
It makes you think about where unexpected scientific breakthroughs can emerge from, sometimes the most unlikely places.
It certainly does.
Yeah.
It highlights the complex and often surprising ways the human body can interact with the natural world, and the unexpected avenues we might discover for tackling even long-standing medical challenges like snakebite.
So where do we go from here? Tim Fried's blood is this potential gold mine, scientifically speaking. But obviously, we can't encourage everyone to start getting bitten by venomous snakes. That's not the answer.
Definitely not.
So what are the most promising future directions for antivenom development? The safer, more scalable routes?
Well, one very exciting area, building on what we just discussed, is the development of what are called recombinant antivenoms.
Recombinant, meaning lab-made.
Essentially, yes. This involves using modern biotechnology, genetic engineering, cell cultures to produce antivenoms, specifically based on human monoclonal antibodies.
Monoclonal, meaning just one type of antibody.
Or a specific cocktail of them, yeah. But highly targeted ones, grown in controlled cell cultures, like CHO cells, which are commonly used in biopharmaceutical production.
Okay, so think of it as precisely programming cells to make exactly the antibodies we need.
That's a perfect way to describe it. Instead of relying on an animal's entire messy immune response, you're manufacturing pure, specific human antibodies.
That sounds much more controlled and, well, a lot less dangerous all around. What are the potential advantages of this recombinant approach?
Oh, there are several potentially huge benefits. First, potency. Recombinant antivenoms have the potential to be much more potent.
Meaning you need less of it.
Exactly. You might need to use smaller doses to get the same or even better level of venom neutralization. That helps with cost and administration.
Okay. What else?
Side effects. Because they can be designed to be fully human antibodies or very close to human.
Not horse or sheep proteins.
Right. They're likely to cause far fewer adverse reactions in patients. Less risk of those allergic responses or serum sickness.
That's a major plus.
Huge. Plus, you can achieve a much higher concentration of the specific active antibodies. Remember, we said traditional ones are only 5-36% active.
Yeah.
With recombinant methods, you can potentially get close to 100% active ingredient, making the treatment much more targeted and efficient.
So going back to your earlier analogy, it's less like a blunt instrument.
And more like a laser-guided missile. Or maybe a set of them, targeting specific crucial venom components.
And what about the cost? You mentioned affordability is such a huge factor for snakebite. Are these high-tech methods going to be prohibitively expensive? Because that often happens with new biotech drugs.
That's always a concern, absolutely. But interestingly, initial cost analyses, looking ahead, suggest that recombinant antivenoms could actually be cost competitive with.
Really?
Or possibly even cheaper than current plasma-derived antivenoms in the long run.
How is that possible?
Well, you're streamlining production.
Yeah.
You're only making the specific antibodies you need, not processing vast amounts of animal serum with lots of irrelevant proteins. The efficiency could potentially outweigh the initial tech investment and developing what they call oligoclonal antibody mixtures.
Oligoclonal, meaning a few specific types mixed together.
Exactly. Carefully selected combinations of multiple, highly specific monoclonal antibodies. That's seen as probably necessary to effectively neutralize the complex nature of snake venom with its many different toxins, but still much more defined than the traditional mix.
That's fantastic news. So instead of relying on animal serum, we could essentially grow these highly specific human antibodies in a lab, potentially at a competitive cost. It sounds like a real game changer.
It has that potential, yes.
Are there any major hurdles to overcome with this recombinant approach? It sounds great, but what are the challenges?
Of course, developing any new medical treatment comes with challenges. It's not simple. One key challenge is the upfront work. Identifying and selecting the most critical toxins within the venom to target.
You have to pick the right targets for your missiles.
Precisely. Out of those 20-50 components, which ones are most responsible for causing the life-threatening effects? We need to make sure we're going after the right ones for broad effectiveness.
Makes sense.
Then there's the manufacturing side. Ensuring that the selected antibodies can be produced efficiently, consistently, and at scale through these industrial cell culture processes. That's a big technical step.
Scaling up production.
Yeah. And finally, there's the regulatory aspect, navigating the approval pathways for these novel treatments. They're considered biopharmaceuticals, which have different regulatory requirements than traditional blood products like animal-derived antivenoms. That takes time and significant investment.
Right, getting them approved for human use.
Exactly. It's a rigorous process.
But the ultimate goal remains that universal antivenom, or at least a very small number of broadly effective ones, right? That would truly simplify treatment.
Absolutely. That's the dream.
Especially in those critical situations where identifying the snake is just impossible.
Yes. A universal or broadly effective antivenom would be revolutionary for snakebite treatment globally, particularly in resource-limited regions where snake identification is a major barrier to timely, appropriate care. It would eliminate a huge amount of uncertainty.
Allow for faster, more reliable intervention, save more lives, prevent more disabilities.
That's the ultimate objective.
Before we wrap up, we should probably touch briefly on the ethics. Not just Fried's actions, but the current methods too. Using animals, keeping venomous snakes.
That's a valid point. There are ethical considerations around the current methods of venom production using captive snakes.
Milking snakes for venom.
Right. The WHO has guidelines, and there's ongoing work to improve animal welfare, refining housing and handling practices to minimize stress on the snakes. It's definitely part of the conversation.
And Fried's actions framing them as this human science experiment, while acknowledging the immense danger and reiterating that it's not the recommended path for scientific research.
Absolutely. His path was extreme and incredibly risky, driven by personal conviction. Modern research strives for ethical, controlled, and much safer methods to achieve similar goals, like the recombinant antibody work we discussed.
Okay, so to recap then,
Yeah.
developing truly effective snakebite antivenoms is an incredibly complex undertaking, a huge challenge. We're dealing with these highly variable, complex venoms. Our current production methods, while life-saving, have significant limitations, side effects, specificity issues, low concentration,
Right.
and we urgently need more affordable, more accessible solutions for the people most at risk around the world.
Precisely. The need is immense.
But the extraordinary case of Tim Fried, despite the risks he took, has provided science with this unique and potentially invaluable resource.
A unique window, yeah. Those highly effective, broadly neutralizing antibodies he developed.
Offering a truly promising new direction, a new inspiration for the future of antivenom development.
It really has spurred a lot of research and thinking.
And the potential of these recombinant antivenoms, these lab-grown human antibodies, and other innovative strategies like combination therapies, it gives us real hope.
Definitely. Hope that we can overcome the limitations of current treatments and significantly reduce the devastating global impact of snakebite.
It feels like we might be on the cusp of a real change in how we handle this neglected crisis.
Let's hope so. There's a lot of smart people working on it now. Which raises an important final thought for you listening out there.
Yeah.
Consider the sheer dedication and the immense personal risk involved in Tim Fried's actions. Was his extreme approach a truly unique, maybe even necessary anomaly in the pursuit of this medical breakthrough? Or does it perhaps starkly highlight the urgency and the often overlooked unmet needs within this neglected tropical disease?
That's a heavy question. Like, did it take something that extreme to shake things up?
Maybe. And it makes you wonder, what other unexpected sources of medical innovation might be waiting to be discovered, maybe in equally strange places? And how should we as a society think about that balance between individual risk-taking and potential global benefit when it comes to scientific exploration?
Something definitely to mull over. Where do breakthroughs come from and what risks are acceptable in chasing them? A lot to think about.